Hello everybody, welcome to today's COSI seminar. We have Professor Aydogan Ozcan from UCLA. Professor Ozcan received his PhD in electrical engineering at Stanford University. He spent a short postdoctoral time there, and then he was appointed as a research faculty member at the Harvard Medical School. He then joined UCLA in December of 2007. Where he's currently an associate professor leading the Bio and Nanophotonics Laboratory at the Electrical Engineering and Bioengineering Department. He holds 17 issued patents and 12 more pending patents. He's the author of a book and more than 200 peer-reviewed research articles in major scientific journals and conferences. In addition, he's a founder and member of the board of directors of the company, Prostia. And of course, he also received many awards, including the 2011 Presidential Early Career Award for Scientists and Engineers, and the 2011 SPIE Early Career Achievement Award. So, welcome to Boulder. Thanks for coming.
Thank you. Thank you very much for this kind introduction and for your invitation. It's a great pleasure to be here. And in this presentation, I'll talk about some of our group work that we've been doing at UCLA with a specific emphasis on telemedicine and what it can do for especially global health challenges that we have today. And at the center of our group's research theme actually lies the fact that we have a very strong cell phone coverage today. If you look at this world map, you can see that cell phones are now being used everywhere. Literally, we have more than 5 billion cell phone subscribers, and more than 70% of these are actually being used in the developing parts of the world. And close to 90% of all of us are living on places that are already covered by GSM network. So this connectivity, when combined with the fact that cell phones are getting much better, much more cost-effective with their architectures, really holds a significant promise to be utilized as a health monitoring platform, as a microanalysis platform to conduct advanced tests in remote locations. One example that I'll be briefly discussing in my presentation later on is actually diagnosis of infectious diseases, in particular, for instance, detection of malaria in blood. This is a problem where most of these countries highlighted in yellow are still in resource-poor regions of the world. So if you had an attachment to the cell phone that could convert the cell phone itself into a microscope that could look at these deformed red blood cells due to the parasite to diagnose malaria, it could really help to penetrate the central hospital that you rarely have in these resource-poor countries to remote villages, even in Africa settings. The same is true for instance, screening of bacteria or pathogens in food and drinking water. All of these problems that are coupled to infectious diseases are actually significant problems for global health. And just to give you the idea, co-infections, HIV, TB, and malaria, they cause more than 4 million deaths a year. And if you add to this number poor drinking water related issues, we're losing more than 6 million deaths, people a year. And all of these problems are actually problems that we could use microscopy to diagnose or to essentially screen. And if we had those working with wherever cell phones work, that could really open up new avenues for us to conduct telemedicine, especially in field conditions. And that's essentially the theme of our research group. And over the last couple of years, we've been providing a new platform to conduct telemedicine, specifically in resource-poor settings where cell phones work, but nothing else could be reliably working. And today I'm going to be talking more about some of these gadgets that you see here that literally convert an existing cell phone, as shown here or here, into a microscope, both bright field and fluorescent. Or if the cell phone doesn't have a CMOS unit or an imager installed on it, we can have standalone microscopes, as seen here, that are powered through USB, that can look at very large specimens in terms of area and volume to conduct microanalysis to look at cells and potentially diagnose disease state. So I'll be talking about this platform, what we've done with it, and what we plan to do with these microscopes. And in my presentation, I'll be referring to the broad umbrella of technologies that I'll be talking about as LUCAS. It stands for essentially a long name, but it's a platform technology that I'll be highlighting throughout my presentation. Before I go into the details of this LUCAS platform, I'd like to actually highlight some specific features of this platform that make it quite different than a conventional microscope, which is quite important, especially if you want to conduct microscopy in resource-limited regions. The first important feature of the LUCAS platform is that it's a lens-free technology. So it doesn't require any one of these advanced objective lenses or the other bulky optical components that you would normally find in a microscope. As a result of that, as you can see in the background picture, these are fairly compact, at the same time cost-effective microscopes. And one unique feature of this platform is its imaging volume. Its imaging field of view and volume is significantly larger compared to conventional designs, which is quite important because most often a disease state is like finding a needle in a haystack. That's why you need high throughput microscopes, and these are actually unique in that sense. Even this small microscope here can deliver actually more than 20 millimeter square field of view with around 0.1, 0.2 numerical aperture, which is quite large compared to a conventional microscope, as I'll be showing you, which typically looks at sub-millimeter square regions. And these microscopes here are actually working based on a unique holography principle, which is not necessarily exactly the same as we've been taught in our lasers classes or holography classes, in the sense that it's based on incoherent or partially coherent holography, which gives us certain advantages. I'll be highlighting them in the next few slides, but makes us quite sensitive to even small-scale features of a bacteria, because most often bacteria, for instance, are index matched in water, and you need a phase contrast ability to resolve them. And holography, the way that we do, gives us certain advantages while also being relatively high throughput. And the final thing about these microscopes are, even though they're holographic by nature, they're quite tolerant to misalignment, in the sense that even if you were to drop one of these to floor, you could pick it up and then it would work fine, because there's literally no alignment in these microscopes that would require a PhD student or someone technical to realign. Lens-free on-chip imaging, which is the major theme underlying LUCAS, is offering us some unique opportunities for telemedicine applications I've highlighted in my previous slide. However, lens-free computational imaging also provides us some unique opportunities for conducting extreme throughput imaging if you don't restrict yourself to the imagers that are installed on a cell phone, for instance, and rather go with large format devices that you can look at microscopy using still a compact architecture as shown here or here, but looking now more than 10 centimeters square. In this case, actually, this is around 18 centimeters square snapshot image of some cells which are fluorescent labeled, and this is the bright field image of, again, around 18 centimeters square field of view. So lens-free computational imaging also has some implications for extreme throughput imaging, which I won't highlight in this presentation. I'll be focusing more to telemedicine and global health applications. However, this type of extreme throughput could be useful for imaging of, for instance, microfluidic devices that specifically capture certain cells or certain types of, for instance, circulating tumor cells from whole blood, so that you can look at an extreme throughput on the order of more than a milliliter of sample solution all at once. So, but I'll again focus to telemedicine rather than on-chip wide field imaging. So the LUCAS platform, because it doesn't use any lenses, essentially it can only image shadows of specimen. And what is quite different between our shadows and a cells or a bacteria's shadow is that the cell itself is semi-transparent, so it casts a unique textured shadow. So if you look at the cell's shadow, it has some fringes, which are actually holographic fringes, and depending on the cell type, you can actually find a different texture, different fingerprint-like holographic shadow. So this is the starting point for a simple imaging platform, which, without any reconstruction, over a very large field of view, which is dictated by the active area of a CMOS imager, which are typically 20, 30 millimeters square, even at the back of our iPhones. With this simple architecture of shadow imaging, where the cell is maybe a millimeter or less away from the active region, we can detect their shadows and their texture in the sense that we can identify which cell is which type through some, of course, training process. If you look at whole blood, in whole blood you'll be seeing mostly red blood cells. These are actually the shadows of red blood cells. Every now and then you would see another one which is coming from the white blood cells. This simple shadow imaging platform can do cytometry based on this fingerprint analysis over very large areas. If you're looking at a benchtop system like this, which is a CCD, and it's got an active area of around 18 centimeters square, then you'll be looking at these holographic shadows over 5 centimeter by roughly 3.7 centimeter. And you would be seeing these diffraction patterns which can enable you to count cells. And again, compared to a 20x objective lens, this platform provides us more than 1,000-fold larger area. And on the top of that, of course, we have some depth advantage so that we can look at a couple of millimeters to understand from their diffraction patterns what kinds of particles or cells are actually located within a volume. This gives us a volume advantage on the order of simultaneously screening around more than a milliliter of volume all in parallel. However, this type of a shadow approach, although quite powerful for certain applications, if you're looking at, for instance, blood, and if you're interested in measuring blood density or capturing of certain specific cells in microfluidic devices, it might be a great solution. However, it has its own limitations because of the fact that cells and their shadows start to overlap with each other. If you look at this blood sample, you can see some shadows here that are isolated and that have these unique texture. However, a region like this, unfortunately, does not tell us a lot just because the shadows overlap with each other. However, this is the time to realize that there is more that you can dig in these diffraction measurements, in the sense that they can actually be processed to reconstruct the missing phase, and then once you have the missing optical phase and the diffraction pattern, you can refocus them to create the image digitally through a holographic reconstruction, which I'll be talking in a second. And this nicely matches to conventional microscope images. So this is the starting point where from these shadows we get more technical and more involved, in the sense that now we can do microscopy with them, even at higher densities where we can't really talk about shadows anymore. It's like a speckle pattern, but even then you can reconstruct and get the images of these cells back. Each one of these cells is on the order of 8 microns here. And again, you can do this with different platforms depending on the sensor that you use. You can do this over a very large area like this, 18 centimeters square. Start from these holographic shadows which are undersampled, reconstruct their phase, and then go back to the object plane over a very large field of view. So whether installed on a cell phone, as shown here, or a standalone unit like this, or a benchtop system, depending on your field of view and portability and resolution requirements, all of these platforms rely on actually partially coherent or incoherent lens-free holography. They are unique in the sense that they don't use even lasers. So this light source here that's installed at the back of this tube is a simple light-emitting diode. The same is true here, and you can also have a light-emitting diode there. So by using a light-emitting diode that's coupled to a very large aperture, which in this case is on the order of 50 to 100 micrometers, we create partial coherence of light, so that by the time that the light waves travel a distance of a couple of centimeters between the LED and the sample, they acquire partial spatial coherence, so that if you ask one of these cells, it's being illuminated by a perfect plane wave with a constant phase, and the next cell would experience something else. But for each individual cell hologram, we have spatial and temporal coherence sufficient to record these holograms. This tuning of coherence, both in space and time, helps us to mitigate a lot of the artifacts that come with laser illumination in typical holography. First of all, there's too much speckle in laser illumination, and that's a noise term. On the top of that, most often when you insert a sample, there will be multiple reflection interference terms coming from the substrate, and that's another noise term. And of course, most often if you're hungry for spatial coherence, this diameter here would be very small, typically on the order of a micron or so. That would have its own challenges to keep this alignment free, in the sense that if you work here with a micron aperture, it may easily get misaligned and then clogged. Working with a very large aperture on the order of 0.1 millimeter is really a big advantage in this sense. Just like any other holographic scheme, this platform is based on partially coherent inline holography. So the reference wave itself is the unscattered wave, and it interferes with the scattered wave to give you the inline hologram. And it suffers from the same twin image artifact, in the sense that because we only record intensities, we have to deal with the twin image problem. And for that, there are different schemes that we can implement. And throughout my talk, I'll be switching from one versus the other. But basically, one way to solve this twin image problem is to go back and forth between the detector plane, which is the hologram plane, and the cell plane, so that you minimize the error. You assume a certain phase at the detector plane and try to recover that phase by going back and forth between the detector and the cell plane. And at the cell plane, you insert an object constraint. That's based on essentially a Garbeck-Saxon type of error minimization algorithm, which gives you the phase to solve the problem of the twin image. And there are other ways which can involve going back and forth between actually the cell plane and the virtual image plane, and you can do some filtering again to converge to the phase. Once you converge to the optical phase, you can go back to the object plane. And one major advantage of using this type of a recording scheme, where the cells are inserted very close to the sensor on the order of a millimeter, whereas the light emitting diode is a couple of centimeters away, is that whatever active area that you have at the CMOS is now your imaging field of view. This one has a 5 megapixel imager here. The pixel size is 2.2 microns. And this is kind of our imaging field of view, where you see holograms across, roughly speaking, 24 millimeters square. It's phenomenally large, and these are the individual holograms, and all of these fringes contain the useful information after phase recovery. And one quick note about temporal coherence. We do not use lasers, so that's why our bandwidths are on the order of 10 nanometer or so. So it's not enough if we were to use a large distance between the cell and the detector array, because some of these oblique rays would not have coherence, temporal coherence, with respect to the background ray. But because we keep the distances very close to the surface of the active region of the CMOS or the detector array, essentially we can live with even a 10 to 20 nanometer bandwidth of LAD without killing our fringes. And that's actually, again, a unique window which permits us to fit in information through temporal and coherence gate and reject the unuseful information, which wouldn't anyway be undersampled by the pixel size, which in this case is 2.2. So in the next few slides, I'll show you some images acquired with this type of a microscope. Keep in mind that the physical pixel size is around 2.2 microns. The field of view is exactly the same as the sensor field of view, which means it's unit magnification. And I'll show you results that are deeply subpixel in terms of resolution, on the order of initially 1.5 micron resolution compared to the pixel size 2.2, deeply subpixel. And later on, I'll show you some tricks that we've come up with to make it sub-micron resolution with the same field of view and same pixel size. And this is, again, talking about reconstructing the phase, which can go back and forth between the virtual and real image plane. Or we can look at it as a simple phase recovery process where it's exactly the same as an error reduction algorithm, where you have this undersampled hologram by going back and forth between the object plane and the phase, the sensor plane, where we update the phase and keep the measurement. We can converge to a hologram phase like this. Once you have both, you can go back to the object plane. So to wrap up this simple parenthesis about the technique itself, all of these gadgets, whether or not they are installed on a cell phone or a standalone unit or on a benchtop system, again, depending on the need of your field of view and resolution, they are working based on partial coherence. And depending on your scheme, you can tune to have more or less coherence so that the information gets in and everything else, like speckle or multiple reflection, goes away. So with this platform, we looked at different kinds of cells to see how relevant such a microscope could be for global health applications. And you're looking at here some blood cells. These are actually holograms of 3 different kinds of white blood cells, like monocyte, lymphocyte, and granulocyte. When you have a 3-part differential white blood count, this is what is measured and separated so that you get all these 3 in different columns. From their holograms, we reconstructed their face. Then you have the object plane here, and these are the subcellular features of the nuclear features of a granulocyte, which makes it, by definition, a granulocyte, and some other. These were actually labeled cells, so that you can see here some Giemsa stain labeling the inner features of the cell. And these are some platelet reconstructions. Once again, these are, relatively speaking, lower resolution reconstructions, giving you around 1.5 micron resolution with a pixel size of around 2.2 micron. Soon I'll show you some higher submicron resolution images. We also looked at some disease states. In this case, we're looking at sickle cell. Sickle cell anemia is actually an interesting problem where a lot of teenagers in the United States suffer with severe pains. Most of the teenagers that are hospitalized are actually hospitalized because of sickle cell pain. And we looked at actually, this is a conventional microscope image, a 4DX objective lens. And the sickle cell itself shows itself as an elongated cell like this. And these are normal, healthy red blood cells. And this is the same region of interest imaged. You can quickly go back and forth from the hologram to the image showing the elongation. Because everything is digital, you can do cytometric analysis and count cell densities so that you can understand if there's an infection. You can understand it by an increase or decrease of the cell densities. And this is showing you how well such a microscope compares against a culture counter, in this case giving you less than 5% error rate compared to a hematology analyzer. And what is interesting is because you have the phase information at the single cell level, you can also estimate the volume of these cells. What we've done here is looking at the phase image and assuming a certain refractive index for a red blood cell, we can understand its volume in femtoliters, and it nicely matches to a culture counter result for the same specimen, patient specimen. We've done the same thing with white blood cells. With less than 10% error rate using a device like this, we were able to measure the density of the white blood cells compared to a culture counter, which is again a commercially available device. One important application of such a microscope and cytometer put together on a digital platform, as simple as the Lucas units that I've shown you, is actually looking at HIV positive patients. For an HIV positive patient, the CD4 lymphocyte is a white blood cell type. It's an indicator of the disease. And for a healthy patient, we have around 1,000 CD4s per microliter of our blood. So once HIV kicks in, this CD4 cell density drops to a couple of hundred. And that's where this density is too low compared to where we are here, more than 1,000. That's where opportunistic infections can be really a pain for the patient. And that's why every 3 months, this density of the CD4 should be measured. Of course, it's a difficult problem because CD4 lymphocytes are difficult to see through a microscope. They look very similar to other lymphocytes. The difference is only at the protein level that surrounds the cell surface. So there are other technologies that can do this task very well, such as flow cytometers, but they are bulky. Even if you were to miniaturize one of these flow cytometers that you can treat as the gold standard for CD4 measurements, the running cost per test is too much. On the order of $5 to $10 per test, which is way more expensive than can be afforded. What we've done is we combined lens-free microscopes that I've shown you with spatial microfluidic devices that have surface chemistry on them so that you can specifically capture the CD4s through anti-CD4 antibodies. Once you inject whole blood to this microchannel, which has a cross-section of 50 to 100 microns, all the rest of the junk will be depleted through the pump, flush, and then just the CD4s will be captured, which then you can image specifically using our field portable microscopes to count the density and the absolute count of these CD4s and other lymphocytes of interest. With a very high specificity and efficiency, thanks to surface chemistry that has been much matured over the last decade or so. And these are actually some patient data for an HIV-positive patient. What you're looking at is a microchip that has printed antibodies. This is an anti-CD4. This is another lymphocyte, CD8. Each one of these spots specifically capture these lymphocytes. And if you zoom in, you'll be seeing their holograms reconstructed and then counted automatically so that we can understand how many cells were captured specifically per site on that chip for a given patient. And these are some zoomed in images for different kind of lymphocytes. In this study, we were looking at the ratio between CD4s to CD8 lymphocytes because that's an important metric for HIV-positive patients. That number should be high, and that's why they have also an interest in another population. And what we've found that with our gadgets, for healthy patients, the CD4 to CD8 ratio was very high, typically more than 2. But once HIV kicks in, that number drops to less than 1. Actually, in severe cases, it dropped less than 0.5 even, and that's where you should start the therapy, the antiviral therapy, before it's too late. So this gives you a good example of how such a simple computational microscope could be leveraging some of these biotechnologies that functionalize through surface chemistry, these microfluidic devices, to bring some more advanced functions to a computational microscope through the specificity of these molecular level interactions. Another important application that we've looked into is looking at waterborne parasites and bacteria. It's a problem where holography has certain advantages. First of all, through its volume. We can look at larger volumes through holographic processes, which is a great advantage to improve your sensitivity. And the other important advantage of holography for this application is we have the phase imaging ability. It behaves like a phase contrast microscope in this sense. If you look at a conventional bright field microscope looking at a water sample that is spiked with E. coli, you would be having a difficult time seeing these E. coli particles. That's why we circled them with red. And these are actually 2 E. coli particles that are being imaged through a 4DX bright field microscope. The same region is now also imaged with Lucas, where you can clearly see the holograms very strong. And that's, again, because of the heterodyne nature of holography, so that small phase disturbances can be converted into amplitude oscillations through the background interference. And that's where we win against a conventional bright field, unless you install a phase contrast objective. These are some other waterborne parasites that we've imaged. These are Giardia and crypto samples. They're hologram captured with a field portable microscope like this, reconstructions, and this is a comparison. Again, the same contrast enhancement is also visible here compared to a 40x, as shown here. In this case, again, this field of view here is 24 millimeters square, and we're looking at a depth of field of around 1 millimeter. So you have there 24 millimeter cube volume screened, which is fairly large because a conventional 40x will be looking at, roughly speaking, 0.5 or less millimeters square. That's why you need to kind of scan or use some enrichment technique. And because of its large volume, we've been able to screen less than 300 CFUs, colony-forming units, per milliliter. And that number can be further improved in terms of sensitivity by another factor of 100 or so if you were to use some simple filtration steps to densify the parasite or bacterial load, which is the typical standard. This was only a study to understand the detection limits of the microscope, not necessarily the filtering before that goes on a water system. So whatever I've shown you so far was acquired with a single LED illumination, very inexpensive low power light emitting diode, which has a bandwidth of around 15 nanometer or so. And it was battery powered, USB powered, and the sample was inserted from this side. And we get a distant resolution on the order of 1.5 micron, which is, again, sub-pixel. Essentially, this 5 megapixel, when you record and reconstruct, is no longer 5 megapixel. It's significantly enhanced because through the reconstruction, we actually get more information at the megapixel count here. And these are some sperm tails. These are 0.5 micron or less in width. And they were reconstructed so that you can do very nice sperm analysis with this platform, which I won't go into the details. So now I'll go to some higher resolution versions of the Lucas platform, which does not give away our advantage in terms of field of view and depth of field. So rather than using a single light emitting diode as shown here, I'll switch to another design here, which has the same CMOS imager, the same field of view, the same pixel size, but has a modified architecture here to really go to sub-micrometer resolution, close to a numerical aperture of 0.5. The key for this is actually to shift the source. So if you shift the source, and if your sample inserted from the site, let's say, is stationary, like in a blood smear or some water sample, then as I shift this by small amounts, like 100 micrometer or so, my hologram, which is static, will be shifting by a couple of microns. And each time I'll be undersampling this same hologram, lens-free hologram, by the same pixel size. Now you can look at this as a way of, based on these undersampled, shifted versions of the same holographic field, how can I synthesize the super result or high resolution holographic frame that was impinging on this sensor before being undersampled. That's actually a problem that has been already solved in photography literature, and pixel super resolution is a terminology that's borrowed from photography literature, or security cameras are already implementing this in the sense that if you have a security camera somewhere in the corner looking at me and I'm standing still, it wouldn't probably resolve my face very well if I'm far away. But if I were to do some small motion like this, left and right, staring at the camera, they would probably love me because they would have a better face reconstructed. And that's the same thing happening, for instance, when they read our car plates with the super result stuff. This is exactly happening here at the geometrical optics level, such that I synthesize, based on this sub-pixel shifts, a hologram now that has an effective pixel size of 300 nanometers, starting from 2.2 microns. And now I can look, instead of a 5 megapixel image, a 0.2 gigapixel, or more than a gigapixel, depending on your image sensor. And now we start to resolve these higher fringes that were normally lost, and they contain the higher numerical aperture information. To get one of these super result holograms, we actually optimize the cost function, which is the mean square error between each one of these undersampled, sub-pixel shifted holograms, and then there's some noise mitigation here, which is looking at the high frequency noise. We don't want the high frequencies to be amplified too much. That's why we put a cost function here to just tune it right so that we don't get a lot of unnecessary ripples. And as a result of this, we can actually resolve sub-micron features. This shows you actually a resolution on the order of 0.6 micron, where the edges of this grating is now clearly resolved, whereas it wasn't for a single lower resolution hologram. And the nice thing is, we don't care if the shift of the source is a raster scan or not. Actually, we don't do raster scans. This was an experimental validation of the concept, which we did raster scan. But if you were to pick, instead of the raster scan, which is shown with blue, some subsets of shift, we would still get the reconstruction from 36 lower resolution. The reconstruction in amplitude and phase, if you were to use 12, is shown here, and if you were to use 5, it's still a distant reconstruction. What is lost here is the contrast, not necessarily resolution. Actually, you can see that the edges are preserved, but the contrast is getting worse as you use less and less. The biggest advantage of this approach is it doesn't give away the field of view, because this is still unit magnification, unit fringe magnification, and you achieve sub-micron resolution over a very large field of view. This is a blood smear that's actually looking at a scale bar of 1 millimeter, and this CMOS sensor has 24 millimeter active area. And if you were to digitally zoom into different parts, you would be seeing the cells reconstructed nicely. Each one of these is a red blood cell. This is a granulocyte, and these are nuclear features of the granulocyte finally resolved, and this is actually a 4DX image. If you see, this is actually a better reconstruction than a 4DX, mostly because of contrast of your projector. And we can do actually very nice reconstructions here. These are some Giardia trepocytes. They have these almost 0.2, 0.3 micron flagella, which are nicely reconstructed here with our microscope. And these are some E. coli particles, which are very difficult to see with a 60x objective lens. That's why we put these markers there, and they're nicely resolved with our microscopes. Well, in a field portable design, it's very simple to do this, to implement this. And that's what I'm going to show you next. So in this black box here, which changes the design to be a super resolved microscope, all we employ is a microcontroller, which is a very inexpensive processor, $3 per piece. It controls 23 inexpensive light-emitting diodes, so that at a given time, only one of the LEDs is on. So the static sample that's inserted from the side from this tray, that's seated above this CMOS sensor, is going to see a single LED illumination, and one at a time, I'll change the LED illumination so that this entire thing is shifting. And as you can see, all the fiber, these are multi-mode fibers, 100 micrometer core. So it's very simple to align because it's 0.1 millimeter in core size. So the LED coupling is done through butt coupling and epoxy fixing done by undergraduate students. So very difficult to misalign. And the rest is just capturing these. And we don't actually use 23 LEDs all the time. Most often 10 is fine. 23 is there because the microcontroller controls 24. So. And these are some images captured by one of these field portable microscopes. As you can see, this is a UCLA pattern written using focused ion beam milling. And compared to a raw lens-free hologram, one of these lower resolution, the super result hologram has all these beautiful fringes, which were normally undersampled, and that's where you get the additional numerical aperture. An important application of this is for malaria diagnosis, especially in field conditions. What I'm showing you here is a blood smear, exactly how it's done in field conditions, where the healthcare worker gets a drop of blood and then smears it on a glass slide with some simple staining, so that you're looking at a monolayer of cells, red blood cells. And the microscopist is required to look at, roughly speaking, 1,000 of these red blood cells under a microscope, high-power microscope, so that they can see these deformations looking pinkish or dark blue, depending on your stain. And this is actually the signature of a parasite. But the problem is, only 1% of the red blood cells show the signature. So you need to look at least 1,000 to be able to see that, say, statistically it's a positive or negative. And unfortunately, with a conventional microscope, that means you have to scan around 100 different field of views. That's a tedious job. That's why a good malaria smear test is done for at least 1 to 2 hours, if it's done correctly. But it's not done correctly like that, because in Africa, the typical time is around a few minutes per slide. That's the reason why we have a lot of false positives. 60% of the reported cases in Africa are false positives. The effect of that is a lot of drug resistance issues because we're wasting our drugs, and the side effects of those drugs are going to be huge for the next generations, and essentially we will have drug stockouts if you continue like this. That's why a digital microscope like this, that can look at a very large area that already contains a couple of thousand cells, and then analyze that at the single cell level would be very important because of this major problem that we have with false positives. That's something that we're very excited about, and we're pushing this kind of microscope a little bit more advanced now to essentially look at large field of views with submicron phase amplitude resolution, and these are phase images which happen to be more sensitive to the early stages of the parasite. So because of the simplicity of this platform, actually on top of super resolution, which you can do by jittering a light source left and right, as shown in this cartoon here. If I jitter the light source left and right in a random pattern, it will help me synthesize a super result, pixel super result hologram, which now contains much higher resolution with an effective pixel size of around 300 nanometers, starting from 2.2 micron. But then, because of the simplicity of the architecture, I can also do an angular rotation and capture super result projection hologram of this cross section, and then the next cross sections, and then put them together. This is exactly how it's done for X-ray computed tomography, except this is limited angle tomography because we can't go all the way 360 degrees. To mitigate this limited angle artifact that would come if I go only plus minus 50 degrees, we also do an orthogonal rotation so that we have now a dual axis orthogonal scheme, which now, instead of looking at a missing frequency cone like this, now the missing frequency cone will be reduced. That helps me improve the axial resolution and remove some of the artifacts that would come. But it's still a problem. What is the optimum scheme? We don't know yet. It's a very interesting question that is open to discussion. But we've implemented just an orthogonal one because implementation of that is relatively straightforward. At a given angle, each one of these excuse me. For a given illumination angle, each one of these holograms that is sampled by a tilted CMOS plane right now is still an inline hologram, because the scattered waves still interfere with the background wave. It's only the sampling issue that is a little bit different now. What we do is we record these distorted holograms, but by squeezing them through knowing the angle, we can actually create a cross-section of the same three-dimensional object seen through this angle now, and we capture the same thing at different angles and synthesize complex images of the specimen at different angles. And then you put them together using a back projection algorithm, very similar to how it's been done in X-ray CT, using a hybrid filtered back projection algorithm. Then we can synthesize tomograms of a very large volume. You're looking at a field of view here of around 15 millimeters square. So the scale bar is a millimeter, and a depth of field of around a millimeter. So we're looking at, roughly speaking, 15 millimeter cube, where we are focusing in, in Z, as you can see here, where these are 5 micron particles, and they're being sliced through this static volume. And they appear, and then soon they're going to disappear. This was on a higher plane, and that's why it still remained there. And these are some static images from the same tomographic data, where we compare our reconstructions compared to, again, a conventional high-NA objective lens. With a high-NA objective lens, you have all these particles in focus. That's understandable. However, all of these particles are out-of-focus particles that are entirely disappearing with the tomographic image as expected. And this is actually a C. elegans. It's a model organism that we know exactly the sequence of its genome, and it's got a couple of hundred neurons entirely. So it's a very nice organism for neuroscientists who especially do drug discovery on this model organism. And you can see different parts of the animal focusing, and then this part is starting to get focused, and it's going to get defocused. Well, there are still artifacts that we can do better, but at least to show the proof of concept, it was fairly good. These are some cross-sectional images of this movie that I've shown you, and its comparison to microscope images and how, if it's defocused, how it looks. So this essentially gives us a spatial resolution of submicron laterally and sub-3 micron axial resolution over a very large volume on the order of 15 millimeter cube. If you want to make this larger, like 50 millimeter cube or 100 millimeter cube, what we'll be losing is mostly the axial resolution, but still you can look at larger depth of field with a reduced spatial resolution, mostly coming because of the signal-to-noise ratio limitations. None of these sensors are actually cooled sensors. They were room temperature, simple CMOS sensors. In that sense, there's a lot of room to improve, especially the CMOS end of our sensors. This was a, relatively speaking, old sensor design. 2.2 micron is not the state of the art. The state of the art today is around 1.2 micrometers. Very difficult to get from Sony because we're a research group, we're not a cell phone manufacturer, and they don't care us as a customer. Even though I say I'm going to purchase 100 of them, they would like to see 100 times 10 to the 4. And because of the simplicity again of the architecture, we can do we can convert the same tomographic design into actually a field portable one. It uses a similar trick as I've shown you before. We use a very similar microcontroller installed here. That is a $3 piece again. It controls 23 light-emitting diodes installed as shown here at the perimeter. Each one of these is butt coupled to a multimode fiber. Very simple butt coupling because the core size is 0.1 millimeter. These are not telecom fibers. And these fibers, as shown here, are actually coupled to a plastic bridge. And only one LED is on here. And as you see, there's a small motion here. That's an electromagnetic motion. So we synthesize super resolution hologram of that angle through this tiny motion back and forth and out of the board. And then after that, we move to the next one. And as you can see, that's how we control the angle. And the sample is inserted from the side so that we do single axis tomography. This is not dual axis, so the resolution will not be as good. And also these are, this is a very inexpensive way of putting it, so it's not as good as the previous benchtop. It has more artifacts, but still it provides us a decent field of view and a decent depth of field. And these are actually some tomographic movies captured with that microscope where, again, we're slicing here the particles. The resolution here is not as good as the previous one, axial resolution. It's sub 6 micron, a factor of 2 worse from the previous one, mostly because it's single axis, and there are more artifacts because of the cheaper design of this microscope. But still, it performs a nice depth rejection ability compared to a conventional single axis microscope objective. So these designs, whether it's on a benchtop like this through shifting of the source and doing some jitter to synthesize super result tomographic images, or a single vertical projection so that you can have a very inexpensive, robust unit here to do blood smear imaging, for instance. All of these were actually using some form of source shifting or rotation. But there are other ways of doing actually super resolved images on a chip in a lens-free configuration. One particular one is actually using the flow itself that you would normally get in microfluidic devices to synthesize super resolution. So rather than shifting the source aperture left and right, what we can do is we can flush within a microchannel a specimen, and it's flowing through. The shadow, holographic shadow, will already be sampled at different subpixel positions. And that is enough for us to take a movie like this. This is a C. elegans, and it's shadow, as you see. There's some undersampling going on, but it's still moving. If we take this movie, we can synthesize a super result version of the animal, even though it's rotating, because we can track rotation and essentially correct for that, so that without any restricting microchannel, very large core microchannels, you can do pixel super resolution and then lens-free imaging on the chip. That's how it's done here. This is a single row hologram of one of these C. elegans bodies, and this is the super resolved one, synthesized through around 10 frames, consecutive frames. And the nice thing is that as the animal is flowing through one end of the microchannel toward the other end, it's going to do some rotation and then some drift. We can know exactly what they are doing, and all we need is a small fraction of those frames to correct for that and then give you a super result image. In that respect, this entire movie, which is on the order of 300 frames, can image the same specimen again and again and again, and reliably, so that all these rotation and shift of the animal can be corrected for. And I'll show you in the next slide reconstructions coming from these 10 frames, these 10 frames, and these 10 frames, so that sequence A, B, and C give us repeatable results, as you can see here. This is a single frame reconstruction, and these are the sequence A, sequence B, and sequence C reconstructed in amplitude images. And you can do the same thing that we've done before, so that as the animal is flowing within the microchannel, you don't need to do super resolution anymore, because that motion already creates super resolution for you. It exactly mimics the example that I've given you, in the sense that if I move across the camera has enough information to super resolve my face. Now we can do that over multiple angles, so that you can do optofluidic tomography, so that you use the fluidic flow to do super resolution and the angular diversity to do tomography. And this way you can synthesize different angles, and each one is reconstructed the same way as I've shown you before, so that you can slice the same animal that was flowing through this microchannel with a tomographic resolution on the order of, in this case, around 5 micron or so. So to summarize, what I've shown you are some of our technologies that we've developed at UCLA to conduct microanalysis using extremely compact, cost-effective, and lightweight units that essentially make up for the lack of complexity through computation. And this is, I think, a timely vision in the sense that most of these CMOS imagers are getting phenomenally advanced. The next generation iPhones and the next ones that are coming are going to be more than 8 megapixels, and that's going to increase our space bandwidth product so that we can now look at the same specimen with the same resolution over larger field of views, or a combination of those where we can improve resolution and keep the field of view the same. And these are some other microscopes that we have installed on the cell phone. This is actually a fluorescent microscope where We flush the sample from the side. This is a microfluidic device, and it essentially looks at wide field of view labeled specimen. This is a field of view of around 9 millimeter by 9 millimeter. We don't count these aberrated regions. And the nice thing here is that the excitation occurs through guided waves. Each specimen that you insert, whether it's a blood sample or some water sample in liquid form, acts like a waveguide, a poor waveguide, but doesn't matter. It still confines light, that is the pump light, in the form of a guided mode, which makes us to use a dollar filter here, a plastic filter, to create these background. So that's a dark field microscope that is created by a $1 filter, mostly coming because of the fact that essentially the excitation occurs through guided waves that are perpendicular to our detection path. And this is actually a movie captured while we're flushing whole blood. So this is a whole blood where white blood cells are labeled. Each one of these green things are labeled white blood cells. There are platelets and red blood cells in the same blood. That's why the contrast isn't that great, because red blood cells that are here are not seen because they're not labeled. However, they scatter light, so the contrast isn't as good. However, by processing 2 minutes of video like this, we can actually count the white blood cell density of the patient, so that it nicely matches to actually a conventional hematology analyzer for this patient. We've done this study and published this already, and it works fairly robust compared to a hematology analyzer. The same technology, instead of using a dummy microfluidic device, as shown here, if you use a spatial microfluidic device that has surface chemistry on it, then it can become like a E. coli sensor on the cell phone. And we've shown recently that we can go down to 5 CFUs per milliliter type of density with a system like that, which is quite interesting, because then you're opening up a lot of applications where with your cell phone you could be able to look at the contamination of your food or water sample with a level that's on the order of less than 10 per mil. So once again, telemedicine has been the focus of this presentation. However, lens-free computational imaging has a lot more to offer, especially for more advanced settings where we don't care about portability and the cost of the sensor. In that case, I don't have to restrict myself to a CMOS imager that is $10 per piece, that is at the back of my cell phone. And I can go with an imager that is made by Kodak, and that has now a phenomenally large area, where in this case you're looking at this is a couple of centimeter wide CCD chip, and you can create a very nice compact imager here, where it's lens-free, but it looks at a field of view on the order of 10 to 20 centimeters squared per snapshot, both in bright field and holographic. So this is the excitation pump, which pumps the blood sample here. That's why it's red. And then there's a vertical source here, which uses this part to conduct holography, so that you can do both holographic and fluorescent of the same microchip. So with this, I'd like to conclude and acknowledge all of our graduate students behind this work and our funding agencies. So, and I'd be happy to take questions. Thank you. The complex amplitude. And so different cells don't share the same phase, but if you look at each hologram, it's a unique phase. So we recovered the phase that's lost at the CCD, or the sensor. And that essentially, you can look at it as a twin image elimination problem, and if you look at it that way, it's best to explain it by going back and forth between a twin image plane, a virtual image plane, and the object plane. And you can see if there's a cell here, at the object plane, the cell is in focus, and the twin image is out of focus. And in the other virtual, it's vice versa. So knowing this, I can say my cell is no longer than this, no larger than this, so I can put a certain box, and then it filters out the twin image artifact, which is much weaker, but distributed a larger scale, because it's diffracting over 2 times the distance. If you do this right, it converges and cleans out this extra information that was overlapping spatially on the top of your object image. That's one way of looking at it. Another way of looking at it is actually forget about the virtual and real image planes, and look at it as a fundamental problem of I have a complex object. It diffracts, and I only sample its diffraction intensity. But there's a phase associated with that, and that phase is not arbitrary, because if I assume an arbitrary phase, if I go back in space, it won't give me a restricted object, because any object that I work with is restricted in space. So then you can converge based on the fact that your object has finite energy and finite space width. It means it's going to converge within all the possible phases that could come with that measured intensity to the one that is correct. That's a great question. For such a large field of view, that is a couple of millimeters by couple of millimeters, it's best done locally, because each time I insert my sample to one of these imagers, it's slightly tilted. It's impossible that it's going to be flat. So the best is to find the autofocus condition local, maybe a millimeter by a millimeter, and then go to the next millimeter by a millimeter, and then find the autofocus so that you reconstruct digitally to take into account that there might be a slope associated with that time that you inserted the sample. We don't do the cell phone computation. Mostly, they're very difficult to hack yet. Android is coming, it's okay, but it's not easy to work with programming cell phones for many reasons, because their operating systems, unlike Android, were not yet, all standardized, so it will be very difficult to find the right person to do that. Today's Android phones that are coming have good processors that we will do some of that processing on the cell phone. However, currently we're using the cell phone for two reasons, either for battery purposes or for connectivity. So that essentially it's going to be helping us to either store power, digitally store or power our microscopes, or essentially transmit some of the data to somewhere that we can communicate back and forth. And that's the current model for any telemedicine system, because the use of the cell phone, whether you're taking the picture of the patient or sending some text about the demographic information of the patient, has to go through a server and then be stored anyway. So I think putting these images along with that other data would be good to close the loop.
How are you getting back and forth between the two mathematics?
The free space function is known. So essentially, I exactly know the phase transfer function of free space.
Oh, so you're actually doing the real propagation?
Yes,
like
digital. Yeah. Exactly, yes. Yeah. Excluding the evanescent rays. Yeah.
I just had a detailed question on one of the slides when you were shifting the small pinhole, and it was showing where it was shifting to. It was sort of a 2D map. It said it was a calculated 2D map of where it was shifting. So this is not something you knew you were shifting?
No, we don't. We don't rely on that because it's not very reliable in the sense that we have a fiber array. Let me go back to that picture again. Okay, let's go here. So these are hanging fibers, and they're not glued, so they're just a line of fibers. And they might, each time, because of the thermal expansion, whatever, they might shift core to core. So we don't rely on that knowledge that they are stacked. What happens is, if you have a cell that's isolated, it essentially shifts a little bit. So you can do correlations between these two holograms to understand how much was the subpixel shift. And most holograms at their central lobes are not undersampled. They're slowly varying. So the most valuable part of a hologram is the tails. Those are the high frequencies, high spatial frequencies that are undersampled. But the central part is already oversampled. And that oversampled region can be a good indicator of exactly subpixel shifts, similar to actually how Palm and Storm kind of can localize things without having the resolution.
Time for one last question.
So the idea of staining image with different fibers, angles, and shift, they random shifts, sounds a lot like what current refractive imaging has looked at.
Yeah, you know, we looked into the papers of David Brady on compressive holography. So we have some compressive work that is going on in our work, but not for holography. We like compressive decoding for incoherent objects better because the forward system is entirely linear. For instance, if I have some slides here, let me show you some compressive work where it's entirely for fluorescent objects. Let me quickly go here, actually. Let me go here. So this is actually a fluorescent image. The raw image coming from the cell phone is not as high resolution, but we use some compressive decoders here to, this is a sparse image, so you know, to get better resolution than the cell phone. So if you look at this cluster here, there are two cells, as you can see with the 10x objective lens, that we reconstruct after compressive decoding. We haven't yet applied compressive decoders like L1 regularized minimizers that are out there for holography because we don't get as good results with them because the forward model is not entirely linear. Because there is interference in the forward model that you can ignore, assuming some conditions hold. But for an incoherent imaging system like this, it's perfectly holding. So we like to image our lens-free or lens-space fluorescent work. We like to work with these L1 regularized type of decoders. But for holography, we're not getting as good results as brute force reconstruction. But there's a lot of nice work done, mostly by David Brady and others that simultaneously published on looking at that exactly a three-dimensional imaging problem that's ill-post, so that you have a three-dimensional set of unknowns, and then a single projection, and you know the forward model, and it can be approximated as a linear system with some constraints on the object function. Then you can you know, approach it as an optimization problem. We haven't published on it yet, but we have published on applications of that on fluorescent lens-free imaging. Thanks.